And so when I was asked to what I want to present, I thought that was in April, present that, and by then we have plenty of results. And as always, the progress is a bit slower than what we expected, but I think I can give you maybe a survey about the topic and some first results, and tell you about the challenges in this field. So if you, first of all, some joint work with 3 colleagues. Niklas is a postdoc in the project about the CWI and in Twente. And Thijs is a former PhD student, and I couldn't find any picture of him in the internet. And so if you read about AI, everyone is extremely enthusiastic. 2 Nobel Prizes this year were awarded to AI, and it all seems what we have to do is to understand transformers, and then we are done. Maybe we can optimize them a bit, and that's it. But one can also maybe think about all these huge challenges that are still there. And we know that deep learning is not very data efficient. You need huge sample sizes, but LLMs, internet scale, and training samples to learn them. And they are also not very energy efficient. So these big tech companies are thinking about building small nuclear power plants for all the energy that they need. And there are other challenges. So, for instance, the way that's run today is the OpenAI. They have the state-of-the-art LLM, but it's still not very profitable business. They make huge losses. I just saw that the new pro version of ChatGPT that will be $200 per month and so on. So there's certainly still room also for completely fresh ideas that go beyond the optimizing existing AI approaches. And so that's a bit the idea, maybe to think about some other directions. What could one do in completely different ways to learn large models? And what could that then be? I mean, given that we have now some understanding of the pros and cons of deep neural networks, what shall we look at? I mean, how shall we do that? And if you think about the evolution of these neural networks, then starting with the perceptron in the 50s, and then 30 years later, we have shallow networks, and another 30 years later, we have deep neural networks. And over time, they seem to more and more resemble the brain, and they're mimicking the brain. So it seems plausible that the future of AI is another step. Somehow the future of AI will be maybe even more biologically inspired. Now, although this suggests the way this is plotted, that suggests that there's a convergence towards biology, I think it's clear that it's not entirely true, because in certain things we deviate more and more from the biology. So in the perceptron, people use the heaviside activation function, which is biologically motivated because a biological neuron fires or it doesn't fire, so it's either 1 or 0. And then because people wanted to use gradient descent methods, and that doesn't go because you can't take the derivative of that. So people smoothed it out, and then they arrived at modal activation functions that were used together with the shallow networks. So that's a bit of deviation, say, from the biology. And then nowadays we have value activation functions, which are even less biologically plausible. Although there's some articles that argue that there's also some biological motivation for value, but I think it's in this respect somehow we also deviate. So it's not entirely clear, and I don't say this has to be the case, but still I think it's interesting to look and to see like what could be the advantages, say, of more biological inspired learning. And in computer science, it's a big topic already. So also in the Netherlands, we have professors there for biologically inspired learning, and these groups around Geoffrey Hinton and Bengio, and so they all published work that. And I think it's a good moment already now for statistics to look into that. I think also if I look back and if we want to learn something from the history, so Hinton, for instance, in the 90s said always that deep neural networks, they will perform much better than shallow networks. Only once GPUs became available around 2010, and so then this could really be implemented. And it was true, but in principle, in terms of theory, one could have checked that out at an earlier stage, since it seems very plausible. And so now we have neuromorphic computing, which tries to put biologically inspired learning on computer chips, and that's not very scalable. But still, we could already try to understand what could be advantages and disadvantages of more biologically inspired learning rules from a statistical perspective. Okay, so that's the framework. So now, when I started with artificial neural networks, I thought this is basically what happens in the brain. And the more I look into this biological literature, you see there are huge differences, right? And they're really much like abstracted away from the underlying biology. And that's for the structure, but also for the way they learn. So in terms of structure, biological and artificial neural network, right, you have numbers and you send numbers through this network, and then you apply these activation functions. Everything is deterministic. On the other hand, the biological network, so it sends point processes, right? So you have point processes, and well, for instance, in the receptive field, somehow you have neurons that they look at certain spots, and if there's light, they're light sensitive, and then they start, say, something which just looks like a Poisson process, and that is then propagated through the biological network. So in that sense, here they are much more stochastic objects, and also the way the information is decoded is very different. So here the information is decoded into the number, and here the information is decoded into the spiking times, the moment when these neurons fire. Okay, so a very different way of decoding information. Still, we have so much results now. Also something I have been looking at in the past 6-7 years, of understanding how width and depth in artificial neural networks, how that contributes to the expressivity and certain abilities of artificial neural networks to adapt to structure. And then one question based on these different structures, whether the same sort of properties hold for biological networks, and whether depth and width, they play a similar role for biological networks. And the structure, that's something we will do in the future. What I will talk about in this tutorial is basically only about the difference in learning. Okay, so the difference in learning. So what's the difference in learning between artificial and biological networks? Well, the thing is, these biological networks, it's widely believed that they cannot be based on gradient descent. Okay, so they can't use gradient descent. Why is that true? Because in order to run gradient descent, it's a non-local method. So you need to know to update one parameter, you basically need to know the values of all the other parameters. So that means if you're in a big network, then there must be some way of exchanging all this information. And the biological network doesn't have the capacity to exchange all this knowledge. So it can only be based on local rules that depend on the state of the parent nodes, the children nodes, and so make that and more. And that's it, right? So, and even like in simple models, think about linear regression models, something I really like. So you see that if you want to run gradient descent for this parameter theta, then to update the jth parameter, you need to take the jth partial derivative of that. And then if you work it out in terms like on the parameter dependence, you see that the update that will depend on the whole parameter vector theta. Yeah, so somehow you have to tell this, if you think about this as a node of the network that corresponds to this jth parameter, you have to send all the other parameter values to that node, because otherwise you could not implement this formula and run gradient descent. And that's interesting because all the theory for AI that we have, it's all based on always gradient descent, gradient descent, gradient descent. And so how can it be that something like, which is so closely related, can not rely on gradient descent and be very, very efficient? Because, I mean, the brain is very efficient. Think about the way we can learn how to drive a car compared to this millions of kilometers of test drive data and billions of kilometers of simulated drive data that is necessary to train self-driving cars. Okay, so all my talk will be basically on the difference in learning. Now, this is just a historical account. This is not something new with the weight transportation problem. Francis Crick, who was involved in the discovery of DNA, he later turned to neuroscience. And then in the 80s, when all this backpropagation came up and also caused a huge hype around neural networks, he had this article in Nature, a sort of opinion article where he says, so as far as the learning process in the brain is concerned, it has nothing to do with backpropagation. Okay, boom, that's it. But what is it then? I mean, okay, so let's look into the backpropagation. Backpropagation consists of two phases. It has a forward pass, it has a backward pass. A forward pass is something where we send the signal through the network and arrive as an output. And that's biologically plausible. So that's exactly what a biological network does. But the problem is the backward pass. And the backward pass, you need to know a lot of things, and you need to do all this exchange of the rate parameters. So if you scrutinize it down to these two passes, then one has somehow the problem is really in the backward pass. Okay, so but then what shall we do? And in the computer science literature, people thought about to replace the backward pass by something that is biologically plausible. Okay, so that's something very characteristic for this whole field, that we don't really know the truth. So basically, we take something which is biologically implausible or so, and then replace the implausibility by something that is more plausible, and then analyze that one. Okay, so but this we don't really know exactly how it works, and that's I think a very good way to get all this started. So what are the methods that are out there that have been proposed to overcome the problems with the backward pass? So one set of methods that can be summarized under the notion of weight or node perturbation. Let's say there's some sort of randomness, right? We saw that these biological networks, they are more stochastic objects, and then just insert some form of randomness without really looking into the biology, and propose a method based on that. And so the forward gradient descent, that's something that I will focus on during this first lecture, is a specific method that you can, that's a specific, that belongs to this class of weight node perturbation methods. And then I think you can understand where all this comes from. Another method that was, I think it's quite well explored, is the feedback alignment. What do we do in feedback alignment? In feedback alignment, we randomly draw weights at the beginning before we start the backpropagation. And we randomly draw weights, and in the backward pass, we always replace the weights that occur in the backward pass by the randomly drawn one. Okay, not the learned ones or so, but just the ones that we picked before we started the algorithm. And so that seems strange that even could work, but people worked it out, and for simple classification problems, it performs quite well. And then for complex problems, it's much worse than the traditional backpropagation method. There's another thing which is called contrastive Hebbian learning. In contrastive Hebbian learning, you think that you imagine that the brain has two phases, and so it knows basically whether it runs the forward or the backward path. And then if it runs the forward path, it's just does the Heppian learning, the biological learning that underlies the brain as we know it. And then you imagine that during the backward path, it changes everything. So it knows that it's now in the backward path, and then it does something like what is called anti-Heppian learning, just basically reverts all these rules. If you think about it, then you can also overcome that. But there's no indication, of course, that there is that there are these two phases and that the brain can know whether it's running forward or backward path at the moment. And also it's not very clear that it could change these laws locally. I think one approach which is very, very promising is predictive coding. And in predictive coding, it's basically doing backpropagation more locally, where the errors are backpropagated layer by layer. And that's proposed by Joshua Bengio and co-authors. Well, in the original version, it says that neurons, there has to be a different type of neurons. Like the value neurons are the ones that basically the ones we know. And then there's a second type of neurons called error neurons that trace all these errors that come basically from the backpropagation step. And now I think this, in the more modern approaches, this is actually all merged into one neuron. And in terms of optimization, it is like, well, there are many different ways to break down optimization problems into local optimization problems by just introducing a lot of slack variables. And that's very close to that maybe effort of ADMM and optimization also. It's very, very close to that approach. Okay, but all of these methods, although they're around for many years and have been proposed by also major figures in computer science, and so very, very, or basically no statistical theory. So all of them can be explored. And that's one thing which if I say, okay, I try to understand a bit more biologically inspired learning, then people say, well, do I need a neuroscience degree for that? No, because it's basically just there, and you can take these rules and try to study their statistical properties. So you don't need to be an expert in that per se to work out, because it's all in the papers, right? The way they write it down, it's very hard for us to understand, but I think you have to translate it into our framework, and then you can just start and try to analyze them. Andre, you want to ask something?
So for grading this up, I guess success from the optimization point of view, you're a stationary pointer.
Mm-hmm.
How do you gauge for these rules, like from the point of view of optimization? Is it a good rule or is it a bad rule?
Yeah, I think today we will go through the forward gradient descent analysis and so on, then you can see it. We have some guarantees, and then So I think all of them, in the end, what they boil down to is those ones, they're all forms of noisy gradient descent. So actually noisy forward gradient descent, you can do that with forward passes only. And then you have to analyze this noisy gradient descent, and you also get to stationary points, or the noise can, of course, push you towards a distribution around stationary points or whatever. Yeah. Okay, so this is the overview. Maybe good, exactly aligns very well with your question. So what are we going to do? So first, I want to look a bit into this forward gradient descent, to some statistical analysis of that. And that's a rule that, just as I mentioned, is just comes out of this idea backpropagation is biologically implausible. So let's replace it by something which overcomes the implausibility of the backward path. And then later, I want to go to even more biologically inspired learning rules that are based on these biological networks. And but they make one big simplification. Instead of point processes, they just work with the firing rates. Okay, so then you make them basically deterministic, and then the deterministic rules on how they can learn. And then probably tomorrow we will talk about spike time dependent plasticity rules. That is even more detailed. And in spike time dependent plasticity rules, those are learning rules that really depend on the firing times. Okay, so that's those are based on the rates, and those depend on the firing times. And then in the end, I want to talk a bit about supervised learning if you for these learning rules based on firing times. Yes, you also want to ask something? Yeah.
So your previous slide, the statistical figuring as optimization theory exists for this type of method, or when you say statistical theory, there is not really a problem.
Okay, so with statistical theory, I mean these risk bounds and convergence rates and so. And for optimization, well, I think it has not been really discussed within the optimization literature. I mean, here you can say this is a part of what we had yesterday, LSA or so. So within that literature, somehow people have understood like also stochastic, or there's a lot of random gradient descent methods and so on. But these specific methods, they haven't been really analyzed. I think my point is from the statistical point of view, where we try to understand their statistical properties, their convergence rates, and so on. Yeah. Okay, so what is now forward gradient descent? Okay, so that's gradient descent. We all know that. So we have a loss function, and then the gradient of the loss, the learning grade, and that's how we update the parameters, so parameter vectors. And now what forward gradient descent does, it's one of these rules where there's some exogenous randomness coming, and so we have in every iteration, we draw a new random vector, psi k. Think about a multivariate normal distributed random vector, and then we update by this formula. Okay, so now you see that you just take a random direction of the gradient, and that's all what we get to see for running this update. And directional derivatives, you can do that via of one forward pass. Okay, so this forward mode automatic differentiation, think about what is a derivative, right? It's you take 2 points of your loss function and you take a certain limit of that, right? So you can definitely see that you can do it with 2 forward paths, and forward mode automatic differentiation allows you to do that directional derivatives with one forward pass. So that's something that you can compute with forward passes only. And so it doesn't rely on this backward pass. Is that now a good method? Well, the problem is that here you have a full d-dimensional vector, which gives you a lot of information. And here you just see this random direction. And of course, you know that in high dimensions, if you draw a random direction psi k, and then this will be likely almost orthogonal to the gradient, right? So that means we lose a lot of information here actually about the gradient. Or you can also see it that instead of a d-dimensional vector, we compressed it into a one-dimensional number. So we already expect that we will probably lose somewhere in the convergence rates in terms of the parameter dimension, say, which is d here. Okay, so what's about the literature? So, for instance, this forward gradient descent set was proposed in this article, all very recent work, 2 years ago, and I think a very nice article, but also hard to read, is that one, which is from the group of Geoffrey Hinton. And here I reproduced something from the open review about this article. It got very high scores, and I think was a spotlight in the end or so. And I just wanted to emphasize this point again. So, I don't want to say anything negative about this paper, but I think one really has to understand if one is in this literature that people don't think about. It's very hard to come up with very good biological reasons to do that. It's really about just replacing biological implausibilities, and that's what they do, and then you try to understand that, right? And so that's just part of how that is.
A machine some formula that isn't zero to coordinate cells, one coordinate.
So in random coordinate descent, that's right. Yeah. So then you would just go through the individual. So you would draw them always the. Yeah. So it's quite close actually. Yeah.
Generally, the psyche have the center with the identity covariance. This is SG.
No. In STD, you take somehow one observation to compute your gradient, and you put that in here.
This is an unbiased estimate of the gradient.
It is an unbiased estimate of the gradient, yeah, but it is very noisy. It's much more noisy than the gradient descent, stochastic gradient descent. Yeah. So you see that later on the rates. So the STD will get the good rates, like the D over K, and this one will get D squared over K. So it's different. Yeah. Mm-hmm. Pardon?
When you said that the forward gradient descent and we computed with one in the form of
right. Yeah.
can you do it? Why don't you need the
So I think the best explanation is if you want to compute a directional derivative at a point, you do this plus h times v minus over h, and let h tend to 0, and that's something you can compute with one forward pass and that one, and so you can see that with two forward passes you can compute that. Sure, and of course you have to take the limit and so on. Yeah. Okay. Other questions on that? Yeah.
what is data is in dimension I think some right?
Yeah. Right.
Psi k multiplied by psi.
Yeah. So you first have to take the scalar product here. That's a scalar product, so that gives you a number, and then you multiply with this vector after.
Jealous by the mathematics. So the dimension math, how will they engine that? You know, jealous means. So I multiplied it.
Okay, yeah. So you multiply a number with a vector, right? So yeah, okay, right. Good. And Harry, yeah.
So are there any analysis about the convergence rate of here? We could be super slow here, right?
So that's what I will do. Yeah. Okay. Yeah. Okay. That's a good idea. Yeah. And I mean, I want to come back maybe to this comparison with STD. Yeah. So STD really would need also the backward pass. So it's and you can get better rates. You will see that. Yeah. So there's definitely a difference. It's a very, very noisy version of gradient descent. STD is also a noisy version of gradient descent, but it has much less noise. Yeah. Okay, so here's a bit the thing you said. So in expectation, you just get the gradient descent back. So it's an unbiased estimator of whether you do, if you do gradient descent, you have here the full gradient. If you do STD, you have just the gradient, the stochastic gradient here. It's depending on what you take, it's an unbiased estimator of that. But you see that if you just integrate out this the noise, then you get a very large covariance. So the conclusion is just very, very noisy estimate of the gradient descent. And now it's so we want to understand the statistical properties. So what we do is we just study that in our favorite model, the linear regression model. Now we want to recover this d-dimensional regression vector theta star here, and then if we write it down with the least squares loss, so the forward gradient descent, it leads to this update rule. What we do is we show that if we say, okay, so these covariate vectors, they should be almost surely bounded by some constant times d. That makes our life much more simpler. And then we tune the learning rate in an optimal way, then we can do that since we consider this as a sort of a negative result. And then there's some, in the beginning, the first iterates, they will be dominated by the initialization. And if we go beyond that, so if we take the number of iterations larger than d squared, then this influence of the initialization is gone, and then we can show that the rate is the d squared over k. And we know this in this problem, the optimal rate would be d over k, right? So we lose a factor d, and that's somehow clear because it's also unavoidable because we compress this gradient vector into one random direction. So we lose a lot of information, so that we somehow need to pay in terms of dimension is clear. So what can we do now about that? Well, one thing we can do is we can say, okay, let's do L updates per observation, right? So instead of just running the forward pass once, we run L forward passes based on every observation. And then there are two things that are going to happen. First of all, we get to see more random directions, L instead of one, but we also get to see more noise, right? So each update gives us a lot of noise. So the thing is, how does that in the end balance out? And the nice thing here is that the noise doesn't matter so much. It's really that we gain, it's basically as the thing that we just have these L random projections instead of one, so we gain in the convergence rate then. Okay, so the result then with L updates per observations is as follows. So in the same setting, and take sigma, say these xi's, they are centered, and sigma is the covariance matrix. And again, we have a learning rate that we have to choose. So it's quite complicated. But I think the important thing is how it depends on the number of repetitions per sample, the L. So we have to scale it in 1 over L, which is somehow clear if you think about the expectation. So instead of having one update, so you do L. So basically you split these learning rates over these L updates. And then we get for the statistical risk of this estimator after seeing k samples and updating L times for every training sample, we get that risk bound. And here I worked out these two terms. One is due to the initialization. So in the beginning, it's very much dominated by the initialization. But the larger the k, at some point, the second term will take over. And then we get as a rate, we get k times this factor a sigma L, which I defined here, divided by a k squared. So basically it will be a sigma L over k. Okay, so that will be the rate for a large number of iterations. And this a sigma L, well, it depends on properties of the covariance matrix. So I was hoping not to have the square here because that the condition number occurs is quite natural. And then here's the influence of the L. So there's the trace of the sigma, which can be as large as d if sigma is the identity. And the larger the L is, the more this part is stamped down until the point where you basically reach here the maximum or the spectral norm of the sigma. And there's another factor d over here, right? So this is the natural factor d in this problem. There's some questions on that. Yeah. Oh, wow. Okay.
So homogeneous.
But what do you mean with homogeneous?
Scale sigma simple.
If you sigma? Oops, sorry. If you replace sigma by some constant, I think then also this thing will rescale, right? Mm-hmm. And so that will come into there as well. So I think overall it should be okay. Yeah. So we in the way we stated in the paper, we just write here a B, and then I would have, this will be the B, I think, and so I think we'll be okay. Yeah. Under, yeah.
Does it change somehow during the over-parameterized setting?
We didn't. We have some plots later. I mean, it allows you to have. So this is the smallest non-zero eigenvalues. So it allows you to have zero eigenvalues as well for the sigma. But we haven't really looked in this over-parameterized setting. Yeah.
You would presume that in the case that if you're in the over-parameterized setting, you try to find, let's say, like the minimum L2 solution.
Yeah.
Always going to help because it's slow regularization. So it actually might improve the ratings compared.
I doubt it a bit. Yeah. A good point. Yeah, maybe you want to ask something. Yeah, please. Yeah, please. Yeah.
And you see people have this observing.
Yeah.
The example, this citing times.
Yeah.
Different iterations for every site.
So whenever you do a forward pass, somehow the thing is that the psi is somehow part of this biological network, which comes in through the randomness of the network. So the natural thing is then to say, okay, every time you run that, you get to see a new psi.
Beat out the few, but you just run one more.
So the same sample, the same training sample, and then you get to see every time a new random direction, which is drawn independently of all the other randomness.
A sigma L is for the magnitude of Z, right?
A sigma L is of the magnitude of what?
Of Z.
No, it can be D squared. If this is the trace of the sigma, can be also D. So it's a D times D metric. So it's
So D. So now this is
So sigma is a D times D metric. So there's some D dependence in the trace, and so it will be that. Yeah. Okay, good. Maybe Martin and yeah.
So to come that your mathematical disjunction by absolute deals with we should take calcium to deals the H-principle.
Yeah, so that's
Feedbacks.
yeah.
The side was in using.
So we okay. Actually, what you should do is you should take L to be okay. That's my next slide. So it's always good that people are very good in predicting my next slide. So my next slide is basically so you should take L to be something like the effective rank, right? And then and that can be small in principle, even in high dimensions. That can be a small number. But I agree, if you just in this case where sigma is the identity, then you have to take L equals to D to go back to the D over K rate. Okay, so that's yeah. Pardon?
Anyway, Joe's ID, you need to get him how to spell about it once.
That's right. Yeah, that's here in this statement. Yeah. But there can be other scenarios. For instance, there can be other scenarios where you then, and we have some simulations. Let me show you maybe the simulations, and then you can see. We also have in our theory something that you basically, So if this is small, then you just need to take L to be the effective rank, and that can be much smaller. But indeed, if you have to compute D forward passes and so on, then you're screwed, because then your computational burden is so large that. Okay, so it's there's one mechanism where you can somehow overcome that, but it comes at the price of running potentially many forward passes, unless you have somehow a nice underlying structure. So I just want, Sasha.
Because we just define the method. And issues alpha K that
Alpha K is the learning rate.
we make this slave people physical cycles, but the next cycle.
Yeah, so alpha K will not depend on psi K. Alpha K, it's complicated. I didn't put it here on the slide. So
What's the best?
I think. Okay.
And IO, right?
So here's the method for the standard method. And now we have improved it by running, and I didn't want to write it down because then it's extremely complicated. I think it's easier maybe just to say it. So if we run, we keep the same data point, the same training sample, and run L updates based on that. And then we get a second index. And then we call these updates theta KL.
No, that's the same, but
Yeah, that's right. Yeah. So I think the thing with the alpha k is it has to depend like 1 over L. That's the important thing. And then it has somehow complicated expression, which again depends on all these spectral norm, the trace, the largest non-zero eigenvalue of sigma. And so it's a bit complicated. So I didn't want to put it on the slides. Yeah, but you can find all the details also in the paper. Maybe I should have put it there. Yeah. Okay. Sure. Yeah.
Message for quantity have the match for the
here, this one. Yeah.
history. So it lets for the online index for y and x the same as for theta in the regular map.
The index for so that's just, I mean, if you would have this without all that, it would be just a stochastic gradient descent, right? And now we do this random projection and
Yj, xk, and we just iterate this with the and size.
so for every training sample, we run L iterates. So think about you augment your data set, and exactly, and that's it. And then you feed that into this FGD method, and that's what we do. Yeah. Mm-hmm. Okay, good. Thanks for all the questions. Let's briefly on the proof. So, I mean, there's some standard methods on stochastic approximation, and so why don't we use those? Well, because there are different things. So, for instance, these two terms that we have due to initialization and the other rate, they're typical. I mean, we looked into that, and they're not decoupled in this standard LSA result. And then we have to deal with the repeated sampling, which causes some dependence, right? So we said, like, if you do run L updates, so then you have a lot of dependence. We would have liked to analyze it also for epochs, where you run through your data set and then go again. But this dependence is even harder to control. But we have some simulation studies on that. And another thing what we do is, which is maybe a bit non-standard, and is the singular covariance matrix that we also allow that one. Okay, so now is to think, now I want to play the same game. Is that biologically now plausible? I think it is quite conceivable that the brain somehow can profit from seeing the same data over and over again. I think also there's a lot of indication now that while you're sleeping, somehow you take experiences from what you experienced during the day and you just repeat it internally again and again. And that has a bit the same flavor as this algorithm, right? Just running that again and again. The same thing. I think like certain experiences are actually at the faster speed and so on, but so the brain does that. Of course, it cannot do that a million times or so, but I think it's somehow conceivable that this repeated sampling for the same training sample improves the learning to a certain extent. Okay. So now some simulations on that. So here what we do is we actually took here the mean squared error and divide it by the dimension. So we want to study the dimension dependent. And then we study a problem of where we take identity metrics, I think, just to see how the dimension scaling is. And what you see is that the forward gradient descent, the plane forward gradient descent without repeated sampling, so that scales really linear in the dimension. And then the other, the SGD and the FGD, somehow they remain stable in the dimension, right? Because we have divided by the dimension, so that means basically they're linear in the dimension, and that means this one is quadratic in the dimension. So a bit like or exactly what the theory also says. Here's maybe that's also related to the other comments earlier. So what happens if you now take as L just the rank of the sigma? Okay, so we also have a bit of theory for that, which I didn't present. but the rank of the sigma can be very small, right? You can have a high dimensional problem, but a small rank. And then you look at this sequence of problems where you increase the rank, and you see that SGD and also the FGD with that many repetitions per sample, you repeat every sample rank sigma times, that rather stays also stable. Okay, so that means somehow because we have divided here, so that's also just linear in the rank of the sigma, which is good. Whereas the standard FGD that would just explode as well in that quantity. So you see that even like with a few, if you have a low rank metric sigma here, then with a few repetitions per sample, and so you can somehow stabilize that. So you don't need to do the D, right? So you can have a smaller number and get the stabilization. You have a question? Yeah.
Well, that's right. So the source of NGD and efficiency is if the standard NGD, you don't really look at all the data points, right? And just have one data point attack.
Mm-hmm. Yeah, so here as well, right?
You still observe something like a whole gradient.
No, it's just individual data points. We can go back again to the formula. So that's really one data point. So you get to see them as pairs here, and then here's you update based on this pair xk yk, which is the case data point. Not so yeah.
The relation problem is it makes numerical descent for.
Yeah, mini-batch sits between SGD and gradient descent, right? It goes into a different direction. I don't think that's right. So here we go away from even, if you think about SGD here and gradient descent here, then the mini-batch sits somehow in between. Or we go to another direction because we have these random projections. So I don't think that even with the repeated sampling, no, I don't think so, because you don't average over different training samples. So yeah.
Sigma does not have low rank, and let's give you any from this. You might as well just put SG and compute each coordinate of the gradient of the full gradient in the form one.
If you do,
you can compute. Using
right, yeah,
with each coordinate of gradients
Yeah, but you get a lot. You think again about the coordinate-wise gradient descent, but here you sample somehow, right? You can sample certain random directions a couple of times and others not, and so they come. And so
if you don't do that, but you do SGD,
yeah,
you could do SGD without doing backwards computations. You can do forward computations only.
That's right, yeah.
Do D computations, but if you do SGD with any qualities, even the same costs will be my
yeah.
G.
So, but then you would have to remember, I think, okay, so the question is, you can, because the full gradients, you can write it as D directional gradients, but that is biologically also not very plausible, because then you would have you would have to remember which directions you already did, and somehow this would have to be aggregated in a good way. And so here there's definitely a difference to that. Yeah. Mm-hmm. Okay, good. Here's another thing where we compare repeated sampling with epochs, and it's basically, just to summarize it very briefly, it's the same thing for a large number of epochs, whether you do repeated L times and then go to the next sample, or whether you run L epochs. That doesn't really matter. So that's the conclusion from that. But we don't have any theory for epochs, because then the dependence is quite difficult. Okay, so this is to summarize the forward gradient descent. So we said, okay, so it overcomes this biologically implausibility of the backward path. So without any adjustment, it has another D in the rate. And so if you somehow do repeated sampling, then you can overcome that. But of course, that can be a huge number of repeated sampling without any structure on the sigma. Okay, now I have a couple of minutes to go to the next topic. Yeah, I think we had quite a number of of questions already. You want me to stop here, or shall I just go on for?
Still like 5.
Okay, good. Yeah, then we go on a couple of minutes. So the idea is then you go dig a bit deeper into the neuroscience literature and you look into methods that are more biologically inspired, and not in the sense that they try to repair backpropagation, but that they really somehow come out of the neuroscience literature. And so now the next thing I want to talk about is these firing rate based local learning rules. And for that, we need a bit of preparation. So in these biological networks, right, so we have nodes, and then these neurons, they emit spikes, and the spikes are sent along these directed arrows. And the spike has an idealized form of a spike is an exponential decay. So it's a potential that has this exponential decay. And so between these, those are weighted, right? So they're weighted by weight parameters as in an artificial neural network. Those are the parameters that are learned over time. And they say how much strength is transmitted, of how much of the energy is transmitted to the descendant nodes. And so overall, if we have that neuron I fires at time tau, and then the neuron J is a descendant neuron of neuron I, then the voltage that the neuron J will receive is of that form. So it has this exponential decay. It starts at time tau and then decays exponentially. Right. Now, so the next question is, what happens now in one of these neurons? How does it aggregate the incoming signal and produce its output? So for artificial neurons, we have activation functions, so we all know how that works. So how does it work now in a biological network? In a biological network, well, the neuron gets to see its these incoming spike trains, and I just plotted 3 of them. So it's about 1,000 or so. And here, just for illustration, those are 3 of them. Then it superimposes them, and at the moment when the superposition, the overall potential, when it reaches a threshold, it's also a bit simplified, but once it reaches a threshold, then this neuron will emit a spike itself. No? So that's also very different from, say, these activation functions, but the closest one is indeed the Heaviside activation function, if you want to link it to artificial neural networks. And that's how it, from its incoming spike trains, how it produces its own spike train. Okay, now these rate-based Heppian learning rules, they propose learning rules that are local, and they depend on the firing rate. So firing rate is not really well defined. So if you think about Poisson processes, point processors, so there would be intensities or so of these neurons that it connects. Okay, so it has it connects neuron I to J, the parameter Wij. So the change of Wij should only depend on these intensities of the neurons I and J, and maybe on something very like dopamine or so, but let's come to that later. And okay, so one of the reasons why that is also not completely realistic is that we know that the brain is very, very fast. So it takes decision after like 5, 6 spikes. So think about a piano player who is really, really fast, right? So it goes very, very fast. And that is not enough that you really like these rate-based models that the averages can kick in, right? So tomorrow we will talk about spike time-dependent plasticity rules where the learning is based on the individual spiking times. But here let's talk about these rate-based models, and they have this, people criticize them for not being entirely realistic as well. And that's a part of the Heppian learning, which just summarizes all learning rules that are based on some neuroscience insight. Okay, so I want to give you now, last 2 minutes, I want to give you some of these rules. These rate-based Heppian learning rules, there's, for instance, the covariance rule that says like how the weight parameter Ij changes depending on the firing rates of the presynaptic, which is the incoming, and the postsynaptic, that's the outgoing neuron. So it looks like a covariance. It's called covariance rule. And there's Oyer's rule. And I think those are the three most prominent ones. And the BCM rule, they're all from the early 80s. And that looks like a long time ago. But I think there's recently, there's some nice literature on that. So you see that's all from, this one is actually from 2024. That's from 3 years ago. So in the unsupervised case, what these methods do, and Oyer's rule is particularly suitable because we know that under certain assumptions, these nodes, they cannot explode, and actually the energy of these weight parameters at every node, they converges to 1, and so it has certain nice properties. And so what it does some form of online PCA. Okay, so if you just have one neuron, then it will extract the first principal component, and you can also manage to get several principal components out of that. But so that's also our understanding of what these things should do, and there's some supporting literature that it does basically some form of online PCA. And we are looking into this as well at the moment. But so what I will do tomorrow, and now I stop. Tomorrow I want to talk about what I think is very exciting is these spike time dependent plasticity rules. And then we get basically back these randomness. Right? So in the intensity models that I just discussed, the randomness is gone because we look at intensities. But once we go back into the spike time dependent plasticity rules, then we get back also this randomness that is also in the forward gradient descent. And they actually turn out to be quite close. And then we will also get what I want to do is I want to present some statistical convergence analysis for spike time dependent plasticity rules. And with that, thank you very much for your attention and for all the questions. Thanks a lot.